Hi. In today's screencast, I want to talk to you about biodiversity. I'm sure biodiversity is a term you've heard before, maybe you have some understanding of it already. But basically, it's a pretty important concept in terms of environmental science, particularly in ecology, because it informs us as to the resiliency of a given ecosystem or species. So let's get into it. By the way, isn't that a beautiful picture? When I was a kid, I lived in the Philippines and I got into collecting butterflies, and I've never gotten over just the amazing, beautiful biodiversity of the order Lepidoptera. Okay, so biodiversity. Basically, the easiest definition I can think of is it's just the variability in living things, like what makes them different from each other. And so it really comes down to two levels. There's the species level, like how many different species are there, but then there's also the genetic level within a species, like how much biologic differences there between given individuals in a given population. So let's start with species level. That's what most of us would think of when we think of biodiversity. So here we're looking at the African savanna, and I see I've got lions, I got zebras, elephants, rhinoceros. Looks like I've got water buffalo, gazelle. So I got all these things that are different from each other. So there's a lot of biodiversity there. There's different species present. But what you need to keep in mind also is that it occurs at a genetic level. So how much variation is there within the genome of a species? So it's quite remarkable if we look at dogs. Okay, so dogs have a tremendous of variability in their phenotype, in the way they present themselves. But they also have quite a bit of difference in their genotype. The genome of the dog species, uh, is that Canis domesticus? I'm not sure. All right, but anyway, there's quite a bit of variability within both dogs and within cats. So there's a lot of variability in their genes, and that gives the species a certain amount of resilience in terms of how it would rebound from some kind of threat to its survival. So that's what really makes biodiversity important, because it provides protection against environmental changes. When the environment changes, the more biological variation there is, whether it's different species or different genes within a given species, there's going to be some members of these populations that are more likely to survive a given stressor. Now, the more variation there is, the more likelihood there is that there's a genetic combination that's going to give this opportunity to survive and continue on. So low biodiversity limits those options. So when biodiversity is high, there's a good chance that some species or some members of a species are going to be able to adapt to this change and survive because they just have the right genetic combination to allow them to do that. But when biodiversity is low, whether it's I don't have that many species or a particular species has, for some reason that we'll talk about in a different class, had their genetic variability reduced, we're less likely to survive these changes. There's just no members in the species that are able to adapt to these rapid changes. And let's face it, the environment changes rapidly, and humans cause a lot of rapid change. So for this reason, biodiversity is a strength to an ecosystem. Now, one thing about humans, okay, I mean, is in our nature to alter the environment. Okay, so one thing that happens with us though is when it comes to growing things for food, be that plants or farm animals, we have a tendency to move towards what we call a monoculture. We're going to plant rice, we're going to plant wheat, we're gonna grow cows, we're gonna grow pigs. We tend to grow like one thing, and we tend to want it to be a certain way, and so we have this sort of inadvertent uh, tendency to reduce biodiversity in order to grow things. Now, here's a classic case: the potato famine in Ireland. It's why my parents. Grandparents moved to the United States. Basically, what happened was, for a long time, people in Ireland, in Western Europe, planted a particular type of potato, and there was one breed they went for because it made really big potatoes. So you have a small patch of land you're going to live on. It made sense to grow this one really big potato because there's more food energy in your garden, so you're more likely to survive the winter. It was great, except one problem: years and years and years of people breeding for one trait, and that is a big fat potato with lots of starch in it. tended to reduce the genetic biodiversity of the crop. So what happened was in 1845, this fungus came along called the potato blight. Probably it just had a mutation in the fungus that made it more able to infect this particular uh, uh, genotype. And as a consequence, in just a few years, pretty much all the potato plants died. Now, this would not have happened in Ireland, died. This would not have happened if there's a lot of genetic variability within the species or a lot of different crops being grown. So had they had, biodiversity either at a genetic level within the potato species or at a species level by planting lots of different things, they wouldn't have all starved to death. But unfortunately, because they didn't have that biodiversity, when this blight came, this fungus came, it wiped out their food source. Lots and lots of people starved to death. There was one of the biggest mass migrations of humanity ever because they didn't have good biodiversity. So let that be a lesson to all of you. Biodiversity rocks. All right, now, turns out measuring biodiversity is a hard thing to do. Okay, but there's a lot of smart people out there, they've got a lot of thoughts. So the two main things I need you to know about trying to measure biodiversity is we want to focus on two properties. One property is called richness, and the other one is called evenness. So richness is basically how many species are present in a given area that I'm investigating. So lots of species means there's a lot of richness, means I have high biodiversity. That's pretty easy to understand, right? You go to the African savannah, there's lots of different animals there. So you know what? Y'all have some good biodiversity. You go to another place and all they have are bison. It's like, well, there's just one kind of animal here, bison. You don't have much biodiversity, okay? Well, evenness is a little bit trickier. So evenness is the degree to which the species are represented in equal proportions in terms of their numbers. So if I count 1,000 individual animals, and 999 of them are horses, and one of them is a gazelle. That's not really a lot of evenness. That's not a lot of biodiversity. Okay, whereas if I have 1,000 animals and 500 of them are gazelles and 500 of them are horses, I have the same amount of richness, but there's more evenness, so there's more biodiversity. So evenness is just the degree to which different species are evenly represented in the overall population of organisms of that particular type in an ecosystem. Like I said, it's a little bit trickier to get hit on, but we're going to work on it today. So I think this picture does a great job of addressing it. Look, on this side here, I have greater richness because I have 1, 2, 3 different species. Over here, I just have 2 different species. So therefore, there's more richness here. But here I have greater evenness because here, if you notice, I have a disproportionate number of butterflies and a small number of ladybugs and dragonflies. Here, I have an equal abundance of butterflies and ants. So I don't have as much evenness here, but I do have, I don't have much richness, but I have more evenness. Now, the question then becomes, which of these two sides represents greater biodiversity? But before we get to that, let's just look at this. Okay, so here what I've got is 2 communities of trees. They both have equal richness. In both cases, there's 4 different types of trees. There's this type and this type and this type and this type. And I have the same 4 types, like 1, 2, 3, 4. I've got the same 4 types in both places. However, if you notice, here I have an equal abundance of each of them, right? I have, like 4 of each of them. But over here, I have a preponderance of this kind of tree that looks like broccoli, right? And I don't have many of these other ones at all. So what that means is over here, 70% of the trees look like this, 12 look like this, 12%, 6% look, but here, 25%, 25%, 24%, 25%. This would have a higher biodiversity than this one by virtue of having the same amount of richness, but greater evenness. All right. Buckle your seatbelts. Don't freak out. I know this looks as scary as can be, but it's really not. Okay, so I would like you to be able to try to put a number on the biodiversity of an area, and people struggle with this for a long time, but they finally came up with a couple working models for measuring it, and this is the one that we'll study. It's called the Shannon Wiener Diversity Index, and the idea is what we're going to do is it's good because it compares. It takes into account both richness and evenness. So let me just walk you through the idea. H is basically the biodiversity index that we're at, and the idea is a bigger number means more biodiversity, and a smaller number means less biodiversity. So it's a way I can compare very disparate environments to try to compare their biodiversities. And this is important because humans, we have this tendency to reduce biodiversity. And if we want to know how much we're impacting it, we've got to be able to measure it. So this is a way of doing that. So what we're going to do is we're going to take the summation of this. We're going to take P is the number of individuals, the proportion of a species, of an individual species, of the total number out there. So in that last example, on the example over here, right, P would be 25%. All right. So, so what percent of the species makes up? But I wouldn't write it as 25%. I write this as 0.25. Right. So I just take that 0.25 times the natural log of 0.25. And I'm going to do that for each individual species. So again, for here, I would do it for this species and this species and this species and this species. And I would come up with a number, an H value. I would do the same thing here. But so here I'd say 0.25 times the natural log of 0.25 plus 0.25 times the natural log of 0.25 plus 0.25 times the natural log of 0.25 plus 0.25 times the natural log of 0.25. And I get a number. But here, I'm going to get a different number because I'm going to say 0.06 times the natural log of 0.06, 0.12 times the natural log of 0.12, 0.7 times the natural log of 0.7, and 0.12 times the natural log of 1.2. And I'm going to get a smaller value for H here than I get here. And that's the value of the Shannon Wiener diversity index. And just here's an example of working our way through this one here. So if I work my way through this one here, I get 0.17, 0.25, 0.25, kind of surprising is that these all worked at 0.25, but I get this value 0.93. If I did it for the other population, I get a higher number. So here's the thing though, even trying to come up with that is you can't really use that unless you can count the species. And that's not easy to do. All right. So here are the main problems with trying to count species in order to come up with a Shannon Wiener diversity index value. And that is how exactly you're going to count them. You go out in nature, what are you going to count? So the problems you come is there's too many species to count. And if you're trying to do it with animals, they have a tendency to move around. So you're not sure whether, is that a squirrel I counted last time or is that a different squirrel? It's hard to tell because of their movement. So people have had to be very, very creative and smart about how to go about doing this. But people have made a lot of advancements in this. So we're not going to get into that much detail, but we need to be aware of the difficulties that might exist in trying to do this. Now, there's two main ways we'll focus on how you go about sampling an area. The first is called a transect. A transect means I just take a straight line, usually by stretching a rope, let's just say, and I just go like every meter I'm going to see what's there. Because the trick here is to try to be as honestly random as you can be. So if you were just to go out yourself, you would have a tendency to go towards things that your brain thinks are more important. So what we find is if I don't have a plan set in place before I go to sample, I get a different value than if I do. In other words, if I say I'm going to go every meter, and at that 1 meter mark, I'm going to see what plant is here, I'll get one set of values. If I just say I'm just going to walk along this rope and count species, I end up with a different set of values. I inadvertently bias my data. And so the idea of transect sampling is to try to help me avoid doing that by just saying I'm going to go every so much distance along this transect line and sample what's there at that point. Now, another way we do it is by doing what's called quadrat sampling. So with quadrat sampling, I'm going to take a rectangular or square area, and I'm just going to say within this area, I'm going to count every single species. Or if that's too much, I'm going to lay out these little intersections, kind of like over the transect line. I say everywhere there's an intersection, I'm going to count the species. It's hard work. I mean, it's tedious work, but it's doable and it provides very valuable data. So what we're going to do today is we're going to do an activity where we use a transect to try to find the biodiversity of a couple virtual forests. Thanks for watching. Let me figure out how to turn this off. What do I do now?